This protocol can be used in the initial phase of drug discovery to identify inhibitors that abolish the activities of EGFR and RAS proteins. The main advantage of this assay is that inhibitory effects of anti-EGFR and anti-RAS therapeutics can be easily visualized in C. elegans.
Begin by spotting 100 microliters of overnight grown E. coli OP50 onto the center of each NGM plate. Allow the plates to dry for 24 hours in a laminar hood, then store them in a polystyrene container. Using a sterile worm pick, gather 10 to 12 gravid adult worms from a previously grown plate and transfer them to a fresh NGM plate seeded with E. coli OP50. Incubate the plate for 24 hours at 20 degrees Celsius. After 24 hours, use a sterile worm pick to remove adult worms from the plates. Incubate the plates at 20 degrees Celsius for approximately 3 days to allow the embryos to develop into gravid adult worms. Collect gravid adult worms into a 15 milliliter conical tube by washing 2 to 4 plates with M9W. Pellet the worms by centrifuging the tube at 450 times G for 1 minute, then decant the supernatant without disturbing the worm pellet. Prepare worm lysis solution by combining 400 microliters of 8.25% sodium hypochlorite and 100 microliters of 5-normal sodium hydroxide. Add this solution to the worm pellet and flick the tube to mix. Observe the lysis of the worms under a dissecting microscope to prevent overbleaching of the embryos. When 70% of the adult worms have lysed, add 10 milliliters of M9W to the conical tube to dilute the lysis mix.